At Range Bio, we're unlocking translational proteomics to accelerate diagnostics and drug development. My name's Brandon Wilson, and I've spent years developing protein assays with my co-founders, Dr. Nico Maganzini and Professor So. And our VP of R&D, Dr. Brachet, has decades of experience across companies like Quanterix and Quest Diagnostics. And we're crazy about proteins because proteins are the superstars of biological function. They're basically mini machines that carry out the critical functions of all living organisms. In the body, protein levels change dynamically over time as genes are expressed up or down based on an individual's health state. Because of the immense importance of proteins, multiple large companies have formed that are dedicated to discovering new protein biomarkers. Using these technologies, academic researchers all over the world have made massive discoveries and deepened our understanding of the proteome. And today, over $60 billion a year is spent on tools just to analyze and measure proteins. But there's a problem. Existing technologies are preventing these discoveries from translating to patients in the places that need them the most. And this is because of three key limitations: throughput, cross-reactivity, and dynamic range. First, existing assays are highly manual and labor-intensive, preventing them from scaling to high-throughput applications such as clinical trials or health screening. Secondly, we know that proteins don't work in isolation. We need to measure panels of proteins, but cross-reactivity limits the number of proteins that can be measured together. And third, perhaps most importantly, is that proteins in the blood span an incomprehensible 11 orders of magnitude. This is a massive dynamic range, the same as the difference between a picnic blanket and the entire solar system. So at Range, we've developed an innovative set of solutions to these three problems. To address throughput, we use barcode mapping technology to convert protein molecules into DNA reporters. This enables us to parallelize thousands of samples simultaneously in a single experiment. Next, for cross-reactivity, we invented the triple lock technology that suppresses off-target binding events, virtually eliminating cross-reactivity. And lastly, for dynamic range, we invented the molecular equalizer. This enables us to normalize protein signals across the entire proteome. So we can dial up the signal from low abundance targets and dial down the signal from high abundance targets so that we can tackle protein biomarker panels that have never been measured before. We put this technology to the test by measuring a panel of 4 proteins that could predict heart disease, but because of their concentration ranges, they could not be measured together on any existing technology. Using the molecular equalizer, we showed for the first time ever that proteins varying over 1 million-fold in concentration could be quantified simultaneously. No existing commercial platform can do that today. As a platform technology, the application space for this is vast. We've honed in on an initial market that has an urgent need for better biomarker measurements. The largest biotech companies in the world run millions of assays every year to support drug development. And we've validated this market with Genentech, who called the potential of the technology a game changer and revolutionary to biomarker analytics. And this market is huge. Genentech's bioanalytical group alone spends over $60 million a year just on protein assays. By our conservative estimations, this represents at least a $3 billion addressable market. And from our conversations with pharma, we know exactly what they're looking for in a product. So at Range, we're packaging these innovations into a reagent kit that can be operated using standard liquid handlers and analyzed using conventional DNA sequencers. Without the need for an additional benchtop instrument, this technology can scale broadly and quickly. And this is a tried and true business model. Luminex sold reagent kits that were operated on standard flow cytometers, and they were acquired for almost $2 billion last year. And pharma is just the beginning. This is a fundamental technology that can be applied to many other markets, including the immunoassay market, the emerging consumer wellness market, and the health screening markets, ranging from cardiovascular disease to cancer to autoimmune disease and beyond. So to recap, recent advances in proteomic technologies have deepened our understanding of the human proteome. But now is the time to translate these discoveries to the real world and improve human lives. And Range Bio is the only company to do this, providing an all-in-one solution for translational proteomics. So thank you.